Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the webinar section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Marc Smith, Lead Product Manager, Process Development from IDBS.
Hi, good afternoon, everyone. My name's Marc Smith. I'm a lead product manager here at IDBS, and we're going to take 50 minutes today to kind of go through how IDBS Polar can help with process development and CMC through process characterization, tech transfer, and into manufacturing. So just very quickly as well onto our safe harbor statement. So today is a demo of our Polar system. Everything you see here from a product perspective is live. Some of the LES forms and things have been configured specifically to do this task, but this is something that we do on a regular basis, and this is something that if you wanted to do, you can do yourself or you can do in conjunction with IDBS and our service teams. So part of IDBS's overall strategy is to be integral to the laboratory, to be in the loop within the laboratory. And what we mean by this is how do we integrate kind of lab systems with the science that's actually happening? How can we as a digital platform support the planning? How can we support the integration? How can we support that execution? How can we provide data sets that are available for searching and easy analytics? And then provide that up to insight layers that can provide you answers to the questions that you're going to ask. And to support this, IDBS Polar, as you can see here, kind of comes with a number of different modules. For the purposes of today, as we're discussing kind of CMC and process characterization, we're really going to be focusing on the kind of these three modules. So the first module is our Explorer. It is an ELN/LES kind of system combined. We've then kind of got an Insights module, which allows us to do advanced analytics and design of experiments and things. And then we've got Control, which is all about process management. That's about managing your parameters and how they evolve over time in terms of criticality and specifications. So as we come back to that lab in the loop concept, we're going to look at it throughout kind of CMC. We're going to start kind of within development, within process characterization. We're kind of going to go through this loop of identifying parameters, designing studies, capturing data, analyzing studies, and updating that process description. The process description now has a life cycle, so we can kind of look at version to version and how each version changes and how the specifications and classifications change with each one. And we can then kind of move up to scale up, doing kind of similar things, and then ultimately into manufacturing, where using our manufacturing edge system, kind of IDBS PIMS, we can then look at inter-batch variation and batch execution and kind of look at that continuous process validation going forwards. And so if I overlay this kind of generic approach to sort of this process validation on top of our software, you can see kind of within the development side, we're really looking at a Polar, and then into the manufacturing side, we're looking at our kind of manufacturing aid solution, which is PIMS. And we've got kind of control, DOE, and insight on here, and then we've got process batch and analytics within the PIMS group. And what we're going to kind of do now is go into these bits of software, and I will show you individually how they work to piece together that process description all the way from the initial kind of parameter definition through to batch execution. So here we are in Polar Control. Polar Control is structured with a site product in mind. So we're really interested in controlling the product and the process descriptions associated with the product. We start out at the top here with some templates. So if you have a template, a process description, you can define them in the system, and then you can use them down below in any of the other kind of sites or products that there are. Within each product, we then have a process definition. Within each process definition, we have a set of unit operations. And for each process definition, we can add a unit operation quite easily by clicking the button, giving it a name, and then we can link this process definition, this unit operation to a specific term in Polar, to a catalog term that's already mapped. This allows us to provide kind of specific lists and unique lists that can be utilized in the parameter definitions later on. In this example, we're setting up a canonical term. So a canonical term in this kind of way means that it's a unique term that's used across the entire system once you get to the data level, rather than the human readable level that you might see up above. If we expand the process definition out, the unit operation out, we can see the different parameters. Each of these parameters has a unit measure and a class, and if we want to add a new parameter, again, we kind of select the dots and click add parameter, we give it a name, we give it a description. In this case, we're going to go for limits only, and then we can look at this in this term linking to the catalog. So that catalog list has now been brought through to the parameter, and we can select something specific that it is. So what this means is you can have different names at that human readable level, but at the data level, you've got a single unique name across the system. You can also kind of determine its parameter class. The parameter classes, again, are they're configured within the system, so any administrators can use them, so you can configure these to match your own kind of business process. Once you've defined that So once you've defined your parameters, we can then move over to the specifications. And for each of those specifications, or each of those parameters, you can add specifications. You can add multiple specifications. You can add the process target. You can add different action limits as well. So again, this is configurable. So if you don't call it an action or a release or an alert limit, you can configure this to whatever you use internally. You can determine the acceptance criteria on here, and you can also put in what method you use to actually analyze these tests. Add a change reason, and move on to the next specification. In terms of the structure of control, it's a hierarchical structure, but again, you can define the hierarchy based on your own business needs. So in this case, we can define a unit operation and a phase. If you utilize the SAD8 compliant models, then also we've got the process and the procedural stage in the system by default. And then finally, you can see a full audit report of everything that's happened in the system. So which user has gone in and what parameters they've changed and what time that parameter was changed. So you get an accurate picture of the version history of your entire process definition as you're constructing it. Defined our parameters that we're interested in. The next step is to then set up to design some experiments. So switching from control, I'm now in Polar Insight, where I can load up the DOE design application. DOE design is a simple way of creating a design of experiment. You can put in the process parameters to test, the process parameters you would like to maximize or minimize. So I'm going to suggest to you what type of design to do. You can select your randomization, your repetition, your center points. It's going to kind of give you hints on how to design an experiment. So you don't have to involve data science if you don't need to. Give it a name, set it to run. It's going to go away into the background. It's going to start creating you an experimental design. So here we can see we've got the standard order planned out, we've got an experimental number, and it's also gave us our process operating conditions, so where we're going to set the agitation, where we're going to set the controller pH for each of those different rounds of experiments. So now that we've designed those experiments, we're going to go back into our ELN advanced product and move on to the next step of the experimental recording. So once you've designed your experiment, the next step is then to run those experiments themselves. And in order to do this, so now we're in our ELN application with our advanced LES module. And so what we're going to do, we're going to kind of go into our LES form here. And this form is going to guide us through kind of designing the series of experiments that our scientists are going to run. So as a study designer, I'm going to kind of come in here. I'm going to think of what product and unit operation I'm kind of look at designing these studies for. And I can kind of move over here to the right and I can look at process locator. Now process locator is going to look into that polar control that we saw earlier. So we can see all of those sites and products that we had. I'm going to look at the bioreactor unit operation and I'm going to import that. And that's brought in all of those kind of those parameters and the specifications and the target limits that's associated with it. I can kind of check them, I can look at them, I can modify them at this point if I want to modify them because I am doing development work after all. If I'm happy with everything, I can then kind of look at doing the next stage of this. So let's match this up with the study design earlier that we did in the DOE app. So at this point, I'm going to import my study design. At least I'm going to import the correct file for my study design. So let's just try that again. So let's import that. And this is going to bring up my study design. In this case, because I'm on one of our internal test systems, I'm going to have to do some manual alignment. So I'm going to align the agitation with agitation and calibration pH with calibration pH. But as you can see here now, this is the study design that we created earlier. We can check it, we can look at it, we can make sure we're happy with it. And then the next phase of this is to really look at expanding on this and joining this with the process parameters to design the experiments that my scientists are going to execute on. Because obviously in the study design, we've only got agitation and controller pH, but actually within the broader process description, you would have other things like stir speed and dissolved oxygen and all of those other great things that we kind of monitor as we go along. So I'm going to kind of go into my run section. I'm going to select my unit operation in my bioreactor. At this point, I can add some additional runs if I want to, and each of those different runs I can assign different parameters to. Once I've done all of this, I can then look at my entire study design. And so this is looking at, for each experiment I'm going to do, what the parameters that I'm looking at, what the target values, what the units, what's the set points, what's the limits. So then what I can then do is I can package all of this up as a work request using our request module and send this off as a study for our scientists to execute on. And for each experiment, my scientists are going to execute on it inside of the LES form. And we do have a 10-liter bioreactor form available, and we do have like an amber bioreactor form available. I'm not going to go into too much of the demo of that side of the LES today because I think that's probably like a call in itself. So if you are interested in seeing more of the LES module, please ask the question, please get in touch after the call, and we'll put you in touch with someone who can kind of give you a demonstration of that. Okay. So now I can look at analyzing that data. So I can go into the Insights application again. So now that my scientists have executed on everything, I'm going to come back to the Polar Insight app. Now, because they've executed on it inside of the LES, inside of the Polar platform, all of that data that they've done has been sent to the database. So if I come into here and I load up the DOE models application, what this models application is going to do is going to bring in the study design that I had earlier, and I can also bring in some of the results from those experiments. So I can see here I've got those experiments, and I've got each of those experiments that I performed, the actual final product concentration as it's coming out from my analytic group. I can then click to run the analysis. This is going to kind of go in, run that DOE analysis. We can look at the target profiles, for example, if that's something you're interested in. We can look at the DOE table in itself, if that's something you're interested in. But the more important one is the significance table. So within the significance table, what we can see here is that based on the dummy set of data that we gave it for the purpose of this demonstration, it's determined that agitation is critical. It has also said controller pH isn't critical, which I wouldn't expect in a real life kind of scenario. But just a caveat that this was demo data that we created specifically for this demonstration. So we're going to look at agitation, and in this case, what next is to then go back to control and update my specification. So now that I know agitation is critical, what I can do is I can now go back into control. And like we said before, that process description now has a life cycle. So I can now go back into Polar Control and update the specification. So now I've come back to Polar Control, where I can see my unit operation, my parameters again. I'm going to go into agitation. And in this case, what I'm going to do is I'm going to go into there and I'm going to update agitation to be a critical process parameter. So now agitation has that change history attached to it. It's got all of those experiments that we performed attached to it. So we can say, actually, the agitation became a critical process parameter based on this kind of set of tests. Once I'm happy with all of my values and I'm prepared and I'm willing, kind of say this is the process description, I can version save this process description, and then I can send it, then I can export it, and then once I've exported it, I can hand it across to the manufacturing side, who, if they're using our PIMS platform, will be able to import this into their system so that we can kind of get kind of cross agreement with those names of those process descriptions. So now I've moved over to a PIMS manufacturing instance. We're going to be kind of working in here. So I'm going to kind of go to my project. I'm going to select a project in here that's empty, and I'm going to import a process definition in here. So if I select and then click import, what's going to happen now is I'm going to select that XLS file that I've imported, and then I can import that over from the development system. In the case of the demonstration I'm going to do today, I'm actually going to import a much larger process description. So we're going to import a process description which is fully filled out with full expansion, N-1 production, and all of those kind of things combined. We've got much more parameters in here. And so this is just to give an example of what a full process description may look like. And you've got to remember all of these things will be defined within development. We'll have executed on them. We'll have took it through that kind of process description version history to create the process description, to understand that design space in order to pass this across to manufacturing and continue execution in manufacturing. So now that our process description is within our manufacturing system, we can start executing on batches. So I've imported my process description into my PIM system. If I needed to, I can then connect all of those parameters up. So if I need to connect to a LIMS system or a DCS system or even something like an OSI Pi system, for example, we can connect those parameters so that we can pull those parameters directly from those systems. And then as you're executing on a batch, or potentially after you've finished a batch, you could then kind of reference that batch within the PIM system as well. So in this case, we're going to look at referencing a production batch. We're going to create a new batch ID, give it a date, and fill in those values as we're kind of going. In this case, we've got product yield, viability of harvest, and then we've got a load of things around offline samples and feed volumes and things like this. So this is just data that you would fill in or capture from manufacturing systems inside of your manufacturing facility. Once we kind of got this into the system, this becomes batch information. A batch in manufacturing is sort of the equivalent to kind of a round or a run or an experiment within the development group. And then we can kind of move on to analyzing this to see if our inter-batch analytics is acceptable. So if I then take us over to the analytics module, now we're in the PIMS analytics module. And the first thing we're going to look at is just an individual plot. So we're going to go over to the production vessel. Let's look at the pH max, and we can see the batch variation on this. So we can see it's being within spec for most of its life and dropped out of spec here towards the end. At this point, we can read the deviation in our QMS system and go for further investigation. If we also need to, we can look at the process capability. So here we can see how well do our specifications fit our process, for example. Do our specifications fit our manufacturing batch? How well do we understand our manufacturing facility? And then just to round it off, we can go into the genealogy, and we can see the kind of the parent-child relationship between those expansion and the N-1 and the production vessels and things like that. So hopefully you can see kind of from a PIMS perspective, this really allows you to kind of control your inter-batch variation and trending. If anything happens, you've got the ability to then go back into the Polar system to assist with your deviation investigations. So where we had that pH deviation, for example, we can go back into Polar Control and we can look at the experiments that went into determining that criticality in those specifications. We can look at the design space and see whether have we deviated far enough from the control space that actually we're in a high risk predicament here, or is this a low risk deviation, for example? And we could potentially really speed up deviation investigation in this case. But what I was hoping to kind of show is how, with something like Polar Control and PIMS, you can get a much better kind of view of your process as it transitions through CMC. So as a CMC leader, as a CMC scientist, you can trust that what you are producing within the development side of the organization is going to work once you reach the manufacturing side of the organization. And then in time, there's a potential here to kind of reduce batch failures, reduce deviations, and all of those kind of great things that come with fully understanding the design space and everything that you're talking about, and really being able to just implement kind of QBD across the kind of the entire CMC lifecycle. So again, just thank you for your time here today. Like I say, I've been Marc Smith from IDBS, and I'd just like to open the floor up for any questions. Okay, thank you.
And as a reminder to our audience, you can go ahead and type in your questions for Marc now. So our first question is, what if we already have an ontology mapping tool across our business? Could we map that or sync it with yours?
I suppose the simple answer is yes. If you've got Polar and you're using the Polar catalog, you can map whatever ontology system you like into the Polar catalog and mirror that. We also have a catalog sync tool, which we can synchronize the catalog to external system sources like SQL databases and things like that.
And it looks like our next question is, as a CDMO, we work with lots of sponsors. What security measures are in place for external collaboration within Polar?
So there's kind of two main ways, probably. I would think we're looking at collaboration. The first is really around in the control module where you're controlling the process description. You can set permissioning at site and product and even to the unit operation level. So, you could share the kind of the definition of the study with the customer, for example. That's probably the main sort of way. And then in addition to that, we've then also got within the Polar LES side, there's also a huge kind of collaboration module in there, which actually allows you to share the data in as close to real time as you as a CDMO would like to share it with the customer, and that customer can be given access to see the experiments kind of as they're progressing. Alternatively, you could kind of create a separate space within the system as well, and kind of put your results in that location and share the results in real time with the customer, for example.
All right, great. Well, it looks like we are running just a bit short on time. So if we don't get to your questions, we'll be passing them along to Marc, who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, what role do you see AI playing in this workflow, particularly on the GMP side?
That's a good. I suppose everyone's talking about AI at the moment. I suppose because in development, what's happening is you're connecting everything up together. So you're connecting the experiment with the sample, with the equipment, with the reagents, and then you're also connecting that with the inputs, which is the process parameters that you're measuring, and the outputs, which is your critical process parameter. So you're getting a really good idea of what constitutes your design space as for your process. What that ultimately means is when we move into manufacturing from a GMP perspective is there's a potential here to actually start predicting what our golden batch will be before we've actually ran enough runs in manufacturing to statistically confirm what a golden batch is. So it's really getting into that predictive capability again. And then kind of just to close the loop on that cycle is once you start doing this in manufacturing and you start to confirm what is your critical process and critical quality attributes, you can then recycle that back into the development side of the organization that states, when I have a new molecule, a new kind of process coming through, based on my historical data, can you predict what will be my critical process parameters going forward?
Great. Marc, we do actually have one other question we might have some time to get to, if you don't mind.
Mm-hmm.
This one says, can experiments be connected to aid data mining?
Yes, they can. Because of the inventory system in the background that connects the assets that you create. So if we take like a generic cell line development run where you create that culture, and then in experiment 1, and that culture is created and then brought into experiment 2, you then get that genealogical tracking between the two. So you can see that, this experiment followed this experiment, which followed this experiment, which followed this experiment. And all of that data can be pushed to as many different data models as you like in the background. So you could have like a sample-centric data model or a culture-centric data model or an experimental-centric data model that basically then allows you to do whatever kind of data mining you like.
Awesome. Well, Marc, thank you so much for your time and insights today. It's greatly appreciated.
Yeah, thank you very much, and thank you for everyone who attended and watched the webinar.
The recorded version of this webcast will be available for on-demand viewing on our website. And as a registered attendee, you will receive a follow-up email providing you with the direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.